Welcome back to G Fun Facts Online. Today, we are going to dive deep into the murky waters of the Amazon basin to discuss a biological miracle that is currently challenging everything we thought we knew about modern medicine. We often look to the future for medical breakthroughs, but sometimes the answers have been swimming in the mud for millions of years. We are talking about a small, unassuming creature that performs a feat of regeneration so complex and so rapid that it seems like science fiction. This creature is the golden apple snail, scientifically known as Pomacea canaliculata. If this snail loses an eye to a predator or an injury, it does not accept a life of partial blindness. Instead, it triggers a biological program of startling complexity. Within just 28 days, a completely new eye emerges. We are talking about an eye that is indistinguishable from the original, complete with a cornea, a lens, a retina, and an optic nerve. This phenomenon is called ocular genesis, and it has catapulted this invasive freshwater mollusk from a simple agricultural pest to a superstar of regenerative biology. For centuries, scientists have studied salamanders and newts to understand limb regrowth. But the apple snail offers something potentially more profound, a blueprint for regenerating a complex camera-type eye, an organ that is strikingly similar in structure to our own. To truly understand the magnitude of this discovery, we first need to understand the subject itself. The golden apple snail is the definition of resilience. It is native to South America, but it was introduced to Asia and other parts of the world as a potential food source. That plan backfired when the snails escaped and became a notorious invasive species. From the rice paddies of the Philippines to the wetlands of Spain, they are often reviled for their voracious appetites and their explosive reproductive rates. They are true survivors, capable of breathing air via a specialized siphon and enduring harsh environmental shifts. However, it is precisely these traits, their rapid breeding, their hardiness, and their genetic adaptability, that caught the attention of top-tier researchers like Dr. Alice Accorsi and Dr. Alejandro Sanchez Alvarado at the Stowers Institute for Medical Research. You see, the scientific community had long focused on traditional models like mice or zebrafish. But mice do not regenerate eyes, and zebrafish have different eye structures. The apple snail presented a unique opportunity because it possessed a complex eye and a regeneration speed that was practically unheard of for such a sophisticated organ. So let us walk through this miracle of the month. How does a snail build an eye in 4 weeks? The process of ocular regeneration in the apple snail is a masterclass in biological engineering. Unlike the slow and often imperfect healing we see in mammals, the snail's body executes a precise, 4-stage architectural plan. The first stage is the emergency response, which happens from hour 0 to hour 24. Immediately following the loss of an eye, the snail's immune system springs into action. Now compare this to humans. In us, a severe injury triggers a fibrotic response. Essentially, our bodies rush to plug the hole with scar tissue. This scar serves as a patch, but it prevents functional tissue from growing back. The apple snail, however, completely bypasses scarring. Within 24 hours, the wound is sealed not by a chaotic clot, but by a clean layer of epithelial cells that prepares the site for construction. This sets the stage for the second phase, occurring between days 2 and 6, called blastema formation. This is the magic phase that distinguishes regenerators from non-regenerators. A mound of white, unspecialized cells begins to accumulate at the site of the lost eye. This structure is known as a blastema. Think of it as a biological blank slate. It is a collection of cells that have seemingly forgotten their previous identities and are waiting for new instructions. Whether these are resident stem cells rushing to the scene or mature cells that have de-differentiated, meaning they reverted to a youthful state, remains one of the most exciting questions in the field today. By the second week, specifically days 7 through 14, we enter stage 3, differentiation and genesis. The amorphous blob of the blastema begins to take shape. The genetic volume is turned up, and cells start to organize. An invagination occurs, forming the optic cup. We begin to see the translucent hint of a cornea and the formation of a primitive lens. Deep within the developing tissue, neurons are being born, destined to become the retina, the light-sensing film of the eye. Finally, we reach stage 4, maturation and wiring, covering days 15 to 28. In this final stretch, the eye does not just grow, it refines. The lens crystallizes into transparency. The retina develops its complex layers of photoreceptors. Most crucially, the optic nerve grows backward from the new eye, seeking out the brain to re-establish a connection. By day 28, the new eye is morphologically complete. It may take a few more weeks for the neural connections to fully mature and for the eye to reach its full adult size, but for all intents and purposes, the organ is reborn. Now the question that haunts every biologist watching this process is simply, how? How does the snail know how to build an eye from scratch? The answer lies in the deep, shared history of life on Earth. researchers performed transcriptional profiling, essentially taking a census of every active gene during regeneration. They found that immediately after injury, nearly 9,000 genes changed their activity levels, but one gene stood out above the rest: PAX6. PAX6 is often called the master control gene for eye development. It is ancient and essential, found in everything from fruit flies to humans. If you mutate PAX6 in a mouse, it is born with small or missing eyes. If you do it in a human, it leads to a condition called aniridia, or the absence of the iris. Dr. Accorsi's team confirmed that PAX6 is the conductor of the apple snail symphony. When they used CRISPR-Cas9 gene editing technology to disable PAX6 in snail embryos, the snails grew up without eyes, proving the gene is indispensable. More importantly, they found that PAX6 is reactivated in adult snails, specifically at the site of amputation. This discovery is profound, because it suggests that regeneration is essentially a rerun of development. The snail does not invent a new way to heal. It simply reopens the blueprints it used as an embryo. Humans also have the PAX6 gene, but in our adult bodies, the on switch seems to be broken or locked away behind layers of evolutionary inhibition. You might be asking, why should we care about a snail eye? Isn't it just a simple spot that detects light? This is a common misconception. The apple snail possesses a camera-type eye, a structure remarkably similar to the human eye. It has a spherical lens to focus light, a cornea to protect the opening, and a retina to capture the image. This is a classic example of convergent evolution. Mollusks and vertebrates split from a common ancestor over 500 million years ago. That ancestor likely had no eyes at all, perhaps just a few light-sensitive cells. Yet, completely independently, both lineages evolved a complex camera eye because it is an efficient physical solution for high-resolution vision. Because the final product is so similar to ours, the apple snail is a far better model for understanding human eye repair than the compound eye of a fruit fly or the simple pigment cup of a flatworm. If a snail can regrow a lens and a retina that looks like ours, the molecular machinery it uses might be compatible with our own biology. Anatomically, the regenerated eye is a masterpiece. But does it work? This is the current frontier of the research. Early studies confirm that the regenerated retina contains all the necessary photoreceptor cells, and that the optic nerve physically reconnects to the brain. However, proving vision in a snail is tricky. You cannot ask them to read an eye chart. Researchers are currently developing behavioral assays to test this. For example, apple snails typically retreat into their shells when a shadow passes over them, which is a standard predator avoidance reflex. If a snail with a regenerated eye reacts to a shadow just as quickly as one with an original eye, we will have functional proof of sight restoration. Preliminary data is promising, suggesting that these new eyes are not just cosmetic dummies, but functional sensory organs. The implications for human medicine are vast. The leap from snail to human is big, but the bridge is built of shared DNA. The inability of humans to regenerate eyes is a major medical tragedy. Conditions like macular degeneration, glaucoma, and retinal detachment cause permanent vision loss because our retinal cells, once dead, do not come back. The apple snail teaches us that the genetic instructions for building an eye are not discarded after birth. They are simply dormant. If we can identify the specific switches that allow the snail to turn PAX6 and its downstream gene network back on in adulthood, we might be able to trigger a similar response in human tissues. Imagine a future where a damaged retina could be induced to grow new photoreceptors by temporarily reactivating a snail-like regenerative program. Or imagine bioengineered eye tissues grown in a lab using the chemical signals identified in the snail's blastema. In conclusion, the apple snail is a reminder that nature is a vast library of solutions to biological problems. For millions of years, this creature has carried the secret to ocular genesis in its DNA, thriving in the mud while we look to the stars. As research continues, the ocular genesis of Pomacea canaliculata stands as a beacon of hope. It challenges the dogma that complex organs cannot be replaced, and suggests that the potential for regeneration may lie dormant within us, waiting for the right key to unlock it. We are only just beginning to read the manual. Thanks for listening to G Fun Facts Online.